colleague Odetta Shafoku, who unfortunately is experiencing some connectivity issues. I'm a materials scientist at EMSL, and I'll be chairing this session on microbial sensors, monitoring nitrogen, carbon, and water cycling in soil. Just like before, we'll host a live Q&A with the speakers following their talk, and we encourage you to post your questions in the chat window. I'm very excited about this session 3. I am pleased to introduce our third plenary speaker, Carrie Masiello, the W. Morris Ewing Professor of Biogeochemistry at Rice University. She will tell us about synthetic biology advances in environmental sciences, biosensors for soil sciences. Dr. Masiello, thank you for joining us. Please feel free to begin as soon as you're ready.
Thank you. I'm delighted to be here. It seems that the video settings have been disabled for me. So I can't, here we go. Thank you. Awesome. So I will start by sharing my screen. Can everybody see my title slide here?
Yes, looks great.
Awesome. Okay. Thanks so much. Thank you very much for the invitation to come here and talk. And so I will talk today. I'm going to give a general overview talk about the role of synthetic biology in the environmental sciences, particularly in the development of biosensors, focusing on biosensors for soils. And I want to start by acknowledging the co-author list here. In particular, I want to acknowledge the last author on the list here, Joff Silberg, who is the synthetic biologist who's the lead in the environmental synthetic biology team here at Rice. And I also want to acknowledge an awesome suite of PhD students present and past, M. Falk, Shelley Cheng, Elena Del Valle, and Lai-Chi Liu, who all of whom have contributed great work that you'll see here in this presentation. So synthetic biology conceives of life as a catalog of genetic software that we can harvest modules from, that we can use to better understand how the world works. And the very simplest application of synthetic biology for this approach is to build microbes as biosensors. And this works because microbes must detect aspects of their environment. And so we can harness the genetic code that they have already evolved to contain to detect properties in the environment that are important, that are microbially important. So the difference between a microbe and a biosensor is that a biosensor has not just the sensing component, but it also has a reporting component as well. And so what you see here is some very early microbial biosensor work that our team did here at Rice. And in this case, we used fluorescent reporters. You can see that, I'll tell you more about these data in a minute, but the thing to notice here is that we use GFP in the middle slide. And then at in the final, we were using a red fluorescent protein as well. So biosensors are microbes that are programmed to report on their environment or on their behavior. And the basic structure of a biosensor is the sensing DNA that senses and code that reports. And so just like a pH meter has a sensor and a reporter, a biosensor has sensor code and reporter code. And in the case of a biosensor, the chassis is actually the microbe. So when we think about what a chassis is, we imagine the chassis is the arm that holds the sensor onto the pH meter. But in this case, the chassis is the microbe itself. And that's an important engineering challenge. How do we work with different chassis organisms? How do we select chassis organisms that are appropriate for specific environments and specific biogeochemical processes? So we can use biosensors for a whole host of problems. We can use them in the simplest manner is as a chemical detector. So we can use them to determine if a specific compound is present in the environment. And this is a property that's widely used inside I wrote here outside soils, but actually in soils as well, folks are using biosensors as chemical detectors. Biosensors can be used as code debuggers. So just as you would put a hello world line of code in your software, you can ask a microbe to report when it's expressed a specific sequence of genetic code, telling you to just, raise your hand if you just expressed a particular line of code in, for example, the nitrogen cycle. Biosensors can be used to report on the flow of genetic material between organisms, and I'll talk a little bit more about that. I'm really excited about the possibilities for that kind of work. And one of the items that's in development, I won't talk more about it here, other than to tell you that there are folks at Rice who are working more on this, is biosensors that report on the memory of the history of their community. So they report not on what they are seeing, but what they have seen in their lifetime, and perhaps even what their ancestors have seen. So biosensors were developed for biomedicine, and that has meant that they typically report visually. And Roger Chen received a Nobel for what we call the fluorescent paint box, these beautiful suites of fluorescent proteins that we can use to understand the behavior of animal and human cells in medical studies. And some of these fluorescent proteins have been ported into soil science questions and are quite useful in some aspects of soil science. And so back to that image that I showed you before, one of the very early pieces of work that we did in thinking about environmental biosensing in soils was we looked at the effects of charcoal on the ability of microbes to communicate with each other. And we used two synthetic microbes. One glowed red when it sent, and one glowed green when it heard. And you can see at the top, the machined version, the fancy machined version of the piece of equipment. The actual original module was 5 pieces of Lego that we stole from one of our children to make the original prints in the agar plates. But we filled, you can see we made troughs in the agar with the mold, and then filled them either with more agar or a mixture of agar and biochar. And this was the beginning of our team's work with acyl homoserine lactones, which are a ubiquitous gram-negative communication molecule. And what we found is that depending on the surface area of charcoal, and actually pH turns out to be an even more important variable than surface area, although you can see in this figure, surface area looks quite significant. Surface area has a very important control on the ability of microbes to talk to each other in soils. And so this begins to explain some of the environmental effects that we see from soil biochar amendment. So some of biochar's behaviors in the Earth system can be counterintuitive. So in some studies, biochar looks like it's shutting off the ability of microbes to talk to each other. And in fact, this is the case, at least in vitro. We showed that this is the case. So why use a biosensor here and not use a GCMS? Well, first of all, it's actually less labor intensive to use a biosensor. But the other reason to use a biosensor in this case is that biosensors have a detection limit that's almost 2 orders of magnitude more sensitive than a GCMS. And it's not always the case that biosensors will be tuned to the right environmental concentrations, but you can imagine that if you're using the detection module in its native organism to detect something that organism needs to know, to a first approximation, it should be detecting in a range that's relevant to microbes in the Earth system. However, so that previous slide that I showed you was not in vivo. It was not in a real soil. And if we want data in situ in soils, there are a couple of options. So we saw some great examples of how rhizoboxes can be used in a talk earlier today to look at the expression of green fluorescent protein in soils. And those applications are yielding, that kind of application is yielding really interesting results. What I want to talk about here is the possibility for using biosensors in situ in whole soils without, with as little modification of the soil structure as possible. And the sensing modules that the synthetic biology community has available have no inherent restrictions on their ability to function in soils. However, fluorescent reporting modules are difficult to detect. So I put here a stock image of soil incubations. And you can imagine that if you were looking at a bulk soil chamber, detecting the expression of GFP, of green fluorescent protein, within that chamber would be challenging, within that mason jar. So we make the case that the ideal reporters for soils, for biosensors, the ideal genetic code for reporting on soil microbial behavior is are biosensors that produce gases. So what we aspire to is mason jar connected to a GCMS, as you see in this figure here, where something happens in the soil and we can watch in real time shifts in microbial behavior, which would appear as peaks in the GCMS trace. And this would work, for example, as a molecule of interest is detected by a soil microbe. So here's our molecule of interest M, detected by a soil biosensor, which triggers the expression of a protein that makes a gas. And of course, the first time that I, as a biogeochemist, began talking with synthetic biologists and they asked, what gas would you like? I said, well, sulfur hexafluoride would be great, and also a chlorofluorocarbon would work really well too. And of course, those are very challenging to work with because they have no natural source. So there's a trade-off here as we think about what gases we use. So we want a gas that is already made by some organisms so that there's genetic code that can be easily snipped out, but it needs to be made by very few organisms so that it doesn't have a large background already in soils. We'd prefer it if it was made by a single gene, so there are not a lot of genes that have to be spliced together. As I said before, we'd like it to be quite rare so that it's not something that's present ubiquitously in soils already. Its feedstock for making its gas needs to be something that's easy to get to. The gas itself needs to not be too sticky, so the gas needs to diffuse out of the soils rapidly without any interaction with the matrix. It needs to be something that's easy to detect. It needs to be a gas that's stable. So ideally, a gas that no other microbe wants to eat. It needs to be a gas that can be sensed and produced across redox gradients. And of course, we all know that's very important in soils because we see such large gradients in oxygen in soils. It would be ideal if we could report on rare members of soil communities. And it would also be ideal if this was a biosensor that we could add to soils and then not add anything else. So we could just put that microbe into a soil sample without adding any media or anything else. So as you can imagine, this is quite a wish list, and we have not achieved everything in this wish list yet, but we have had some successes, which I'll tell you a little bit more about in a minute. And I'd also want to note that as we think about using gas reporters, we want to remember that there are single gas problems and two gas problems. So there are going to be experiments that I'll show you that require one gas, and then experiments where you have to have microbes that are making two gases simultaneously. So there are lots of options for gas reporters because bacteria make lots and lots of small molecules. And what we've started with are the methyl halide transferase family. And methyl halide transferases make methyl halides. So they make methyl bromide, methyl chloride, and methyl iodide. And they make them non-specifically. So MHTs, when they have lots of bromide, will make methyl bromide. MHTs will make methyl chloride if instead they have lots of chloride. So we do not yet have methyl halide transferases that are specific to chloride, bromide, or iodide. We are not working with methyl iodide right now because of toxicity issues. But in theory, methyl iodide, if you gave an organism with methyl halide transferase in its genetic code, lots of iodide, it would make methyl iodide. So MHTs have some of these properties that we wanted. They're encoded by a single gene. The substrate is something that's quite common. It starts out with S-adenosylmethionine. Halides are easy to get into a soil. And I'm not going to talk about working under very low halide, soil halide conditions, but M. Falk, who will speak not the next talk, but 2 talks after this talk, will talk about how low can you go in terms of halide addition to soils. Methyl halides have a very high vapor pressure. They're not terribly sticky. They're not produced in many soils. They are persistent in soils, and they're relatively easy to measure. So the first test that we did was to ask if MHTs could produce methyl halides and exceed background production. And in many different strains of organisms that we've engineered, so Pseudomonas stutzeri, Rhizobia, Aceinorhizobium, and then Schwannella, have all been able to produce many orders of magnitude more methyl halides compared to a wild type organisms. And then, of course, in thinking about soils, we needed to know, can organisms that are programmed to produce MHT, programmed to produce methyl halides, can they produce methyl halides under realistic soil water properties? And this documents serial increased dilution of soil, going to 0 to a quite dry 33% water holding capacity. And the reporter here is methyl bromide over colony forming units. And that's not our favorite denominator. And I'll talk a little bit more about that when I talk about 2 gas and 1 gas problems. But the answer is yes, methyl halides can be produced down to as low as the lowest we've tried, which is 33% water holding capacity. So the first result I'll tell you about is monitoring horizontal gene transfer in soils. And this is a 1 gas problem, which is why we started with this problem. And personally, I think this is one of the most interesting tools that has come out of the environmental synthetic biology team here. I'm really interested in how the properties of a matrix affect the ability of organisms to share genetic material. I think this is a fundamental question about how a planet impacts evolution. How does the matrix that life exists in shape the trajectory of evolution in a fundamental, a very basic manner? But then also it has some pretty significant environmental remediation and environmental hazard capabilities as well. So this is a 1 gas problem because we want the signal to grow as the population of transconjugates. So organisms that have the ability to produce methyl halides. As this population grows, we want to see that signal growing. So how do we do this? So we do this with 2 strains of E. coli. So 1 strain of E. coli is the recipient, and it does not have a conjugative plasmid. The other strain is the donor, and the donor strain has a conjugative plasmid that has the capability to produce methyl bromide. However, that capability is repressed in the donor strain. So the only way you get any signal is when the recipient receives that plasmid from the donor organism. So each time gene transfer occurs, we see an increase in signal. So the magnitude of the signal reports the magnitude of genetic transfer. And so the first proof of concept we did in soils here was to show that it is in fact possible to use this technique you can monitor horizontal gene transfer using gas reporters instead of by pulling out plating and growing transconjugates. So this plot here on the right is the concentration of methyl bromide versus the concentration of transconjugates. So these organisms here that have received the plasmid. And this lovely linearity shows that this tool works. And so this tool is ready for testing the effects of changing nutrient status on the ability of plasmids to move in the environment, the effects of environmental chemistry, the effects of the effect of the Birch effect on soil wet-dry cycles, on horizontal gene transfer. And so just as an aside, why would you care about horizontal gene transfer if on properties other than, as I said before, just you might want to fundamentally understand how the existence of a matrix affects evolution. You might be interested in how rhizobia transfer nitrogen-fixing plasmids when they're not in symbiosis with plants and when they are in symbiosis with plants. You might be interested in antibiotic resistance and the transfer of antibiotic-resistant plasmids among bacteria throughout soils and engineered environments. What are the properties of an environment that speed or hold back antibiotic-resistant transfer? And you might care about how soil texture and soil moisture impact all of these. And so I'm actually really excited about this tool. I hope others are interested in this as well. Please let me know if our team can do anything to get you started. All right, so that was a 1-gas problem. We also have 2-gas problems, and this is a microbe's perception of its environment. So the challenge with these is that the population of a biosensor can grow, and it can also shrink with time. And so the sensor population can grow and shrink over time. And this is problematic if you have just 1 gas, because you can't tell the difference between lots of organisms that are emitting a little bit of a signal or one organism that's emitting a lot of a signal. And so I'm going to return to the example of the effects of soils on the ability of microbes to detect these ubiquitous gram-negative communication molecules, acyl homoserine lactones. So many aspects of both the carbon and the nitrogen cycle are coordinated by AHLs. So how microbes choose to make biofilms, gram-negative organisms, when they make biofilms, they coordinate that through conversations using AHLs. Spore-forming organisms have been shown to coordinate the production of spores with AHLs. Rhizobia are in conversation with each other and with plants during the process of plant infection, and there are 5 different AHLs that are simultaneously involved in that conversation. This is an example from the marine environment here. The production and destruction of sinking particulate organic matter in marine systems is coordinated by acyl homoserine lactones. And then finally, one example that's not on here, there's really strong evidence that the acyl homoserine lactone communication system has been horizontally transferred to archaea, to methanogens, and methanogens, aspects of methanogenesis are also managed using this communication system. So it's a really important system in the environment, and we wanted to know what is the impact of the matrix? What's the impact of the environment on the ability of microbes to manage these conversations? So for the 2-gas problem, the solution here is to use 1 gas to monitor growth. So there's 1 gas that is always on. And in this case, it's this green gas, ethylene, represented here as ethylene forming enzyme. And then methyl halide transferase is conditional. So methyl halide transferase is only expressed when a particular condition is met. In this case, the detection of an acyl homoserine lactone. And then what we do is, instead of reporting the gross concentration of the conditional signal, methyl bromide, we report the ratio of the conditional signal to the constitutive signal. So methyl bromide over ethylene. And there are a bunch of reasons why ethylene is not perfect. And we'll talk about that a little bit more in a couple slides. We were, however, able to use this paired system to look at the effects of the presence of soil on microbes' ability to detect different chain lengths of AHL. So we started with a short chain length AHL, 3-oxo-C6 homoserine lactone. And we looked at the ability of microbes to detect this signal in liquid and in soil. And so this top black line is the detection in liquid. And the second line here is detection in soil from Kellogg Biological Station, because we had a lot of soil from KBS in the lab at that point. So we started with a 3-oxo-C6, and there are some changes here in the detectability, but it's not huge. However, when we move to the C12 homoserine lactone, so a longer chain compound, and I apologize, the chain length did not increase here, but the chain should be longer for this compound. We see a very significant matrix effect. So almost 3 orders of magnitude shift in the detectability, a 3 order of magnitude reduction in the detectability of this signal in the presence of soil compared to the presence of liquid. So this tells us that different aspects of the AHL communication system in soils are regulated differently by soil texture and grain size. So in this area, the next step that we really want to work on is more gases. And so that ethylene gas is not perfect, as I said before. So the ethylene forming enzyme only is able to make ethylene in the presence of oxygen. And as everybody here, I'm sure, knows, soils are not 100% oxygenated all the time. In fact, lots of the biogeochemistry questions in soils that we're interested in occur only in low oxygen or no oxygen conditions. So this is something that really needs to be addressed. And so one of the areas of development is halide-specific methyl halide transferases. So I mentioned before that the methyl halide transferases that we're working with right now are not specific. So if you give them lots of bromide, they make methyl bromide. If you give them lots of chloride, they make methyl chloride. And that's been a grand challenge for almost a decade for the team here, is to develop methyl halide transferases that are specific. So one that would make methyl chloride and one that would make methyl bromide. So I'm going to move next to talking about some work that's in prep that is very close to submission from our group, done by Elena Del Valle, or led by Elena Del Valle, looking at how individual soil properties impact the behaviors of soils. And we really need tools that allow us to get somewhere between living soil and the Petri dish. So as anybody who has attempted to interpret results of soil experience, natural soils are incredibly complex, and it's very, very hard to compare different soils because you can't entirely control the difference between 2 field sites. You can't guarantee that only one thing changed. And the artificial soils that we're designing are tools that work in this space between the Petri dish and living soils. So they're a controllable model. They're not an exact model, but they're a controllable, manipulatable model that allow us to look at how soil properties affect microbial behavior. And so starting with monodisperse quartz. So it turns out you can get quartz from fracking companies that has extremely tight grain size control. So you can really choose your soil texture very tightly for artificial soil construction. You can add minerals to shape the content, the clay content, the iron presence or absence in the soil. The pH can be manipulated. We aggregate using a couple of different methods. So we have a couple of different options for aggregation. We can aggregate using organic carbon or aggregate without organic carbon solely through wetting and drying cycles so that we can control for the effects of aggregation differently from the effects of presence or absence of organic carbon. and then add a growth medium, because if you're doing microbial experiments, it's nice if they move on a more rapid timescale. So the first thing that Elena's work has showed us that I'm super excited about is we can use artificial soils to understand what soil properties are additive and what soil properties are emergent. And so, as I said before, these soils are made from a base of silicon dioxide. So here are some examples of them. So these are synthetic soils, and they're white because they have that silicon dioxide base. So what you see in this plot here is starting. So this is the ability of a microbe to detect AHL again in a liquid culture. And so it's great at detecting AHL in a liquid culture because it's not seeing any effects of absorption and there's nothing to prevent diffusion. And then each line here is stacking another soil property into the artificial soil. So the artificial soil, as this figure goes in this direction, properties are being added to match a mollisol that we happen to have in our lab as well. And so the most important property in triggering microbial behavior to move the farthest toward natural properties is particle size. So getting soil grain size right is the single most important step. So particle size, adding mineralogy is another significant step, and then getting the pH right. All three of these, particle size, mineralogy, and pH, all bring the artificial soil closer to the natural soil. And what we can take from that is that these properties, grain size, mineralogy, and pH, these properties, at least for this system, are behaving additively. When you add them, you get something that gets closer and closer to the natural system. Now you'll notice I did not talk about this brown line here, which is organic matter. And so in this case, the model organic matter used was chitin and xanthan. And when that's added, it drives the system away from natural, from the natural soil. So going from this soil and adding that one more layer of organic matter takes it away from the natural soil and properties. And this feels, we don't have a great explanation for why this is, but it does feel truthy to those of us who work with organic matter, that organic matter adds complexity to natural soils, and that we might expect organic matter to behave as an emergent property and not as an additive property. I'm going to give one more example here of some biosensor work that we've done. Again, this is work by Elena Del Valle, talking about the effects of organic matter on microbe-plant communication, which again are counterintuitive. So in a paper published with Johannes Lehmann's group last year, Elena and Tara Webster showed that as organic matter in soils increases, the ability of microbes and plants to talk to each other drops. So organic matter dials down that conversation. It mutes the conversation between plants and their symbionts. So before, when we were looking at the acyl homoserine lactone biosensor, I told you that it was 100 times better at detecting AHLs compared to a GCMS. In this case, an HPLC, which is what we would use to detect naringenin, the flavonoid used, one of the flavonoids used in this conversation, the HPLCs and the biosensor respond in about the same way. So this is a calibration curve for the HPLC, and then this is a similar curve for the biosensor. So they both have about the same detection limit, and they both have about the same response range. Oh, they do, however, tell you different things. So the HPLC tells you how much of the material is there, and the biosensor tells you how much a microbe can detect. And those two are not always the same. And that's why when Elena did this work, she chose to run both an HPLC and a biosensor to see if there was a difference. And spoiler, there isn't in the case of this particular compound. So in these experiments, the biosensor, the signal, the neurinogen signal was incubated in soil for 24 hours. And then the supernatant was removed and added to HPLCs or biosensors for detection. And the soils used here varied in organic carbon content from 0.5% up to 7%. And you can see both the HPLC and the biosensor show that the detectability of neurinogen, that signal, is attenuated. It drops in the presence of soil organic matter. And so I'm going to end here and then just put in a plug for the talk right after Trent's talk, which is given by M. Falk, and she'll talk about a biosensor to monitor bioavailable nitrate in soils. So she'll talk about our group's progress developing a nitrate biosensor, and then also progress thinking about what needs to happen to bring these soil biosensor incubation conditions as close as possible to natural soil conditions. And so as the biosensor, if you want a huge population growth with a really robust signal, you want to add we end up creating non-natural conditions, but there are trade-offs in creating more realistic conditions. And Emma's going to tell you about some work she's been doing to really optimize that. So I want to end by acknowledging the students in both the Silber group on the left and my group on the right, and our sources of funding.
Thank you so much, Carrie, for the great talk. Amazing advances in soil biosensors. So we have a few questions in the chat. The first, the pH question, you kind of hinted about in the beginning, whether the biosensors are pH dependent. Maybe you can tell us a little bit more about that, like pH range, or how would you consider pH in their applicability?
Okay, hang on. I'm looking for the, here we go. Is biosensors, okay, our pH dependence of biosensors. Well, so they're microbes. So they function, the biosensor itself is, it's got a chassis organism, and that organism has a natural range that it can live in. And so if you were to pick an acidophile organism, then it would function best in a very low pH environment if you built on an acidophile chassis. If you built on a halophile chassis, you'd have an organism that functioned really well in highly salty environments. So it's going to be a function of the chassis organism.
Great, thank you. The next question is about mineralogy. Are you characterizing particle size and mineralogy of the soils throughout the wetting and drying cycles, since mineralogy will change as a result of dissolution and precipitation?
Oh, I see. And then we often use AQDS as a proxy for organic matter. I apologize. Sorry. I don't know what AQDS is.
Yeah, I'm not sure.
We do characterize the particle size of the soils initially as we produce them. In the artificial soils, we have not. Well, we put them through wet-dry cycles to create, thank you. Wet-dry cycles to create aggregates, and then we have aggregates, and we characterize the physical shape of the soils through at that point after they're done with wet-dry cycles. So we have not done any experiments with the artificial soils where we've subjected them to wet-dry cycles after making aggregates.
Okay, thank you. There's a comment, love this work. I'm reminded of experiments by Stark and Firestone exploring the range of soil moistures over which diffusion limits rates of nitrification, and lower soil moistures under which microbial metabolism becomes limited itself by desiccation stress. Would love to see these techniques applied to discern such tipping points in drying, wetting cycles for various microbes orchestrating.
This is a really thoughtful and interesting suggestion. And I especially like the beginning sentence here. I'm reminded of experiments by Stark and Firestone. And I just want to take a moment to say that Mary Firestone has probably done it already. So all of us who do anything having to do with soil microbiology, the first thing you should do is check, did Mary Firestone's group do it? Because she's really been such an incredibly prolific and thoughtful scholar. But this is a great question. I would love the opportunity to work on a problem like this. We don't have anybody working on this right now, but yes.
Great. I was wondering about, okay, so next question. Okay. Yeah, it's another comment on the pH dependence, that in microenvironments, it's dependent on the pH. I think you already answered the pH dependence. I was wondering about your outlook on potentially deploying these sensors in the environment. So, all over the country or the world. Is that one of the visions?
So as I was building this talk, I thought about whether or not I should put the ethics slide in at the end, and I didn't. But I think that's an important question that we need to think about as a community. Like, how do we feel about putting genetically modified organisms into soils in an uncontrolled way? So that's an important conversation, and I think it's important that we have that conversation now and not after somebody has done it. So I guess I would turn that back to you. What do you think? What do you think about deploying engineered microbes in soil environments?
That's a great question. Maybe it requires discussion with the science community, right, and the as well as the OE.
Yeah, I mean, some folks have said, well, it should be okay because the leak rate is probably only 1 in a million organisms will escape into the natural environment. And then, well, a million, when you add microbes to a culture, working with 1 million organisms is not a unusually large number. So that line could actually be saying you're guaranteed that some of these organisms will leak into the environment. So I think we have a lot to talk about, and we should talk about before we do any deployment.
This also sounds like some additional studies exactly to that point would be useful to show that, justify that it's okay or not. There's a comment that there are controls for this type of thing being developed by Dion Antonopoulos at ANL.
Yeah, and as soon as GMOs are out there, you can't take them back. Better be safe than sorry. Yeah, so in fact, there are synthetic biologists who have thought about or thinking about how do you put kill switches into organisms so they're guaranteed that they die? How do you encapsulate organisms? And the challenge is that you have to be really, really sure that that there will be no leakage. Like none, like zero, nothing, no leakage ever. And that's a very high bar.
Yeah, so that also may require some additional sensor development.
But there's so much to do in the lab. I don't think we need to take these. I think we can do some really cool science in the lab. And when we're done, pop those guys in the autoclave.